everybody. Welcome to the Silver Lyming. I'm Anne Desjardins, your host for this wonderful podcast that I brought together to educate people more specifically about vector-borne illnesses, as well as to raise hope and really let people know that not everything is so dark and desolate when it comes to the vector-borne illness family and spectrum. I have been through it myself. It took me 7 years to get an accurate diagnosis, and it took me another 7 years basically to fix myself and regain my health and actually come out even further and as a better person than when I went into this situation. But that's a really long time in terms of lifespan, and I just really want to do my best to help people to understand different ways they can look at healing, different ways they can think about keeping themselves and their families safer. And today I have a really wonderful guest with me. I love all of my guests. We're all wonderful and have something to contribute to this conversation, but he focuses specifically on babesia and fungal diseases, malaria, and dengue fever. And I'm just very curious to learn more about this topic because it is something that is becoming more prevalent in our environment, which I think he will also speak to a little bit about that. And many people don't really get when we're looking at vector-borne illness, that could really be anything from ticks to mosquitoes that are spreading these illnesses. We often have the idea that it's Lyme and Lyme alone. And in reality, there is a whole set of co-infections that people can contract, and babesia is one of those. And I gratefully never tested positive, and I don't think that I ever had it, but when I look back on my journey, I think there are some times when potentially I do or did have it. I know air hunger is one of the more prevalent symptoms of it, if you will, or maybe one of the more recognizable ones. I think that's really what I'm trying to say there. And I did have that a few times, but I was diagnosed with a litany of other tick-borne infections. Supposedly all came from a tick, but I do think actually I might have been bit by a mosquito. And I just want people to understand a little bit more about the line between those two things. And my guest today, again, is Dr. Geoffrey Dow. He's the CEO and president of 60 Degrees Pharmaceutical. And he really has at least 20 years of experience in infectious disease research and product development, including a strong background in tropical diseases. So we think about babesia as more of that and similar to malaria. He co-founded 60 Degree Pharmaceutical in 2010, where he has been instrumental in advancing drug development, securing FDA approvals, and managing regulatory affairs for products like tafenoquine, Arakoda for malaria, and with a rich history of leadership at the Walter Reed Army Institute of Research, Dr. Dow has developed a keen understanding of the challenges involved in creating effective treatments for infectious diseases, including babesiosis. Ooh, that's a long tongue twister at the end there, a lot of infectious and diseases. But at any rate, I really do appreciate you being here with us and your time, and I look forward to this hopefully enlightening conversation for people. If I could ask you again just to share with us a little bit more about how you became the CEO of 60 Degrees Pharmaceutical, what put you in this direction? Was it something personal that sparked an interest for you? Was it just something that you happenstance came across and were like, this is something good for me to spend my energy towards?
Oh, Annie, thanks for the opportunity to speak with you. So I'm I guess a parasitologist by training. So originally a research scientist, now more of a business guy and clinical trialist, but originally in the lab. And my experience at Walter Reed was in some ways awesome, but it also highlighted some of the challenges in moving products forward in a kind of in more of a institutional government setting as opposed to the private sector. And so I needed a change is probably the simplest way of describing it. And then I'd had an exposure to tropical health and some of the issues with diseases of poverty in Indonesia and other places as a kid. And that was why I worked on malaria to start with. And so when we started 60 Degrees, it was with the idea of moving forward products that were focused on the unmet medical need, but could also be partnered with an indication that was profit generating, and then sort of focusing on the idea of dual use products. So we started out in dengue, which is where that part of the bio came from. And then around 2015, we licensed a product called tafenoquine from the Army. And tafenoquine is the active ingredient in our commercial product, Arakoda, which we eventually got FDA approval for in 2019. So the approved indication is to prevent malaria in travelers. So if you're going to Africa or Central America, where the malaria is common, it's typical to take a daily or a weekly product to prevent malaria while you travel. That's the standard medical advice from CDC. And that's because there is no vaccine that will protect people who don't have any immunity from malaria from the disease. So there is a vaccine that has been developed in an African context for young kids who get exposed to malaria all the time as they grow up, and so there's some immunity already built in. Whereas you and I, if we get bitten by a mosquito and get even one bug in our blood system, you're going to have fever and then the start of a potentially pretty serious illness. So we had gotten tafenoquine or Arakoda approved, commercially available right at the end of 2019, and then the pandemic basically didn't allow us to do a formal product launch like we'd hoped. But then maybe in 2020 we started getting calls from patients trying to locate Arakoda. And it was usually with a description of a chronic disease, and it was never very specific at the time. It was usually symptoms that could be Lyme, could be babesiosis, could be long COVID. And slowly we started to see our sales go up, even though we were doing no commercial outreach really at all. And then some articles started appearing in the literature, and then there was a case series. And so then we sort of paid attention. And maybe 2 years ago, we made a decision to switch our conference strategy to go to an ILADS conference,
Okay.
as opposed to the normal. And that was a completely, for me, a career-changing event. And I heard for the first time patient accounts of the journey that you go through before you get a Lyme diagnosis. I heard about the challenges that physicians have who themselves are open-minded about these things, just some of the professional challenges, some of the issues with trying to follow evidence-based medicine, but the evidence base for some of the standard treatments is poorer if you compare it to a disease like malaria, for example, where there's pretty good treatments, they're short, there's lots of clinical trials supporting them, and partly it's an issue of a lack of resources, and partly it's an issue of this long-standing divide in the medical community between more mainstream folks and then some of the alternative providers. And so that's how I came to vector-borne disease. So we have basically made a decision as a company to focus most of our R&D efforts on a series of 3 small clinical trials in acute, and then hopefully soon in chronic disease as well. where we're trying to essentially validate the patient experience with chronic babesiosis, but doing it in a systematic clinical trial and research-driven way, so we can then take the data and go to the FDA and get a label change, which will allow much broader prescribing to treat babesiosis, where at the moment there are drugs available, but there's no formal labeled product that can be used without other issues.
Okay, thank you for that. And so then I think I'm a little bit confused. Is Arakoda more for prevention or for treatment after contraction?
So the mechanism is the same. So malaria, tafenoquine and Arakoda are unique amongst antimalarials in that they kill every life cycle stage of the parasite. So in malaria, that's into the liver, and then you have a quiescent form in the liver, which can reactivate. And then you have the blood cycle infection, where the parasites multiply in your red cells, the red cells burst, and then they keep multiplying. And that's what causes the anemia and fever that's associated with malaria. And babesia is analogous in the blood stage part of it. It invades red cells, it multiplies, the red cells burst, and in the acute stage of disease, it causes anemia and fever. But the difference between most of the malarias and babesia is with babesia, the infection can last a long time, even when the initial acute phase of the disease has been addressed with atovaquone or azithromycin. And the way we think tafenoquine works is it targets those parasites in the red cells via the same process, which is basically an induction of oxidative stress inside the red cell, and that kills parasites, whether it's babesia or malaria. So what we think happens with tafenoquine is it knocks down the acute infection, and then it suppresses or eradicates the persistent forms that can last for many months after the initial infection. So that's our hypothesis for how it works. So in the context of traveling, you've got drug on board already, which means the parasites don't survive following a mosquito bite. They don't make it through the liver, and then even if they do, they get into the bloodstream and the drug's already there. And then for treatment of either malaria or babesiosis, where the parasite's already started the invasion cycle, then you hit them with drug and it kills them and suppresses an active infection.
I see, thanks. And so then when it hits the liver makes the red blood cells, and is that how it's proliferated that way? Or no, it's...
No, it actually, with malaria, the parasite, after you get bitten by the mosquito, the parasites travel to your liver and actually invade your liver cells.
Mm-hmm.
And they have a cycle of replication in the liver, and then they burst out of the liver into the bloodstream and then invade red cells in the bloodstream.
Okay, thanks. And then would that be why with something like babesia you have air hunger, is because the red blood cells are bursting, so the oxygen is not carrying through the body, or no?
So for acute disease, where you can have shortness of breath and anemia and fever, those things are definitely related to the parasite burden, because you've got a lot of parasites, a lot of red blood cells bursting, and usually the acute symptoms are relieved as the parasites are getting cleared by whatever drug treatment you're on. So those things correlate very well. I don't know what causes air hunger, and I don't know what causes, people have described it as bone pain in people with chronic disease. I don't know the answer to that.
All right, thanks. My head was just like, oh, I wonder if that's why that works like that. But not necessarily one of the questions that I, I had thought of beforehand, but just in that discussion, it made me think a lot about that. And then when people are contracting something like babesia, is that more from ticks, more from mosquitoes? Where does that come from?
So this is a tricky area, and here we have to separate what we know and has been proven from what we don't know and people and folks speculate about. So what we do know is if you go out into the woods and test ticks for babesia, you'll find it. Not in every tick, but in a decent amount of them. So it's reasonable to think that the geographic range of ticks that carry those parasites might be broader than we think it is, or there might be different types of ticks that can carry babesia that infects humans. And people have reported case studies of types of babesia that are not the typical ones. So, we usually think about Babesia microti in the northeast as being the main species that causes disease, and it probably is. But then there are case reports of Babesia odicoli and other Babesias that we don't know what the species is causing disease as well. I've never seen a paper where anyone said they had found Babesia in a mosquito or a vector that wasn't a tick.
Okay.
We do know that there can be transmission through bodily fluids, so near transmission from mother to fetus, from organ donors through the blood donation system prior to the introduction of the new testing, and probably through blood contact as well, although there's less. The evidence for that is mainly from the veterinary literature.
Interesting.
I'm more cautious about saying never or no to things than I used to be, just because we see how damaging that can be. But at least what we know from what's been reported in the literature, it's thought to be a tick-borne disease.
Okay, thank you. You're also touching on another one of my questions was how many kinds or strains of babesia are there? You're obviously let me know that there's definitely more than one kind. And is that more of a regional breakdown than where the different types come from, or it just depends on the actual tick species that's carrying it?
So the kind of the dogma is it's northern Midwest and Northeast problem, and 99% of it is caused by Babesia microti. And I personally believe that in the northern Midwest and the Northeast, it probably is Babesia microti that's causing most of the human illness. But then, there are papers that have shown in case reports other species. We know that Babesia divergens is one that's contracted in the US. So I prefer to think of it as a disease rather than as a species. So I think the incidence of symptomatic babesiosis is much more geographically widespread than the Northeast and the Midwest. And with our own market research, we've seen diverging ranges from how low it could be to how high it could be. And I think there's a lot more research that needs to be done to track both which ticks and where have babesia, and much more surveillance of blood samples to see what kind of low-level persistent infections are out there and where, because I don't think we know the answer to that.
Okay, and I am gonna say I think I know the answer to my next question, but is this just a US phenomena, or is this also like Lyme disease around the globe?
So we know there's babesiosis in Central Europe and Northern China. And I suspect that I suspect it's sort of a bit like the US, which is we know what we know, but we don't know what we don't know. And it's probably more geographically widespread across Eurasia than what the literature would suggest. And of course, we know from the veterinary literature that there's strains of babesia or closely related parasites like malaria that infect a whole lot of different animals all over the world, often with lifelong infections. And the that and this is me expressing my personal belief. I suspect that our lack of knowledge is a big function of a lack of resources and attention. And if we had focused on babesiosis with even a fraction of the budget or research energy that we do with malaria, it would look like quite a different disease, both in terms of geographic spread, the types of treatments available, and our understanding of chronic versus acute disease.
And how good is testing for this? So again, coming out of the Lyme world, like I know how inaccurate Lyme testing can be. How is it with long-term babesia? Is this something that's hard to see in the bloodstream once it's there, or does it have a protective film, a biofilm around it that it can kind of hide? Is it like a shapeshifter like Lyme, or no, it's readily detected?
So based on my knowledge, I think it's a parasite burden question. So when you have acute disease and you've got a 2 or a 3% parasitemia, so when I say that, I mean 2 in every or 3 in every 100 of your red blood cells are infected. That's easy to see on a blood smear. And any technician who knows how to prepare a film and look down a microscope will pick it up. But if you're on drug treatment for 3 days, your smear goes negative almost immediately. So if you think about the context of if you have a summer illness, you feel a bit sick, but you didn't get to your doctor in time, if they only do a smear, they're not going to pick it up. Right, so that's the first issue. Then we go to standard commercial PCRs, so from LabCorp or LabQuest or Mayo. And I'm just referencing those three because they're known to be well validated, and they run thousands of other tests for Americans every day, health related, that people believe the outcomes of. But those cease being able to detect babesia very soon after you recover from clinical symptoms. Once the parasite burden's gone down, their detection limits, maybe 1,000 parasites per milliliter of blood, which means you've still got a lot of parasites ticking over, and those tests will screen negative. And then for the blood bank, where you have by definition asymptomatic people, and you're trying to eliminate them transferring just a few parasites through some blood to a donor, they have an exquisitely sensitive test which screens down to around 3 parasites per ml. And so And what we've seen since FDA approved that blood bank test is that the rate of transfusion transmitted babesiosis cases has dropped off substantially. So what that tells you is that there's a mainstream medical community belief that low-level asymptomatic infections actually have something real biologically going on because they can transmit through the blood system, and they've developed a test to rule that out that's very exquisitely sensitive, but it isn't commercially available for patient care. So the short answer to your question is, if I personally knew I'd been bitten by a tick and I had fever and chills and malaria-like symptoms, I'd be very skeptical that I didn't have babesiosis if my PCR came back negative. And I don't know what the consequence of not getting treated and having a low-level parasite burden is. It could be that that's what leads to or contributes to chronic illness and chronic fatigue. Or it could be that, and this is the kind of current mainstream medical view, that it's not very important. And as a kind of professional in the field developing drugs, I'm sort of in between those two points of view. I have to operate to run a clinical trial to prove that something's real. But all the things I've heard from patients and what the quality of the diagnostic test suggests is there's an awful lot we don't know, and we need to be open to that. So that was a pretty long-winded answer to your question, but hopefully it was helpful.
It is, and it just sort of leads me back to the same place that I would be with Lyme and the standard 2-tier testing, and that it's maybe not as robust as it should be or could be, and you just have to really monitor more so on symptom set as opposed to sometimes actually what the blood results would maybe share with you. And that's where it gets a little bit more of a slippery slope, because again, if we don't have the certified tests to show that we have something, sometimes it's a little bit challenging to get medical care because practitioners are hesitant to prescribe things without an actual clinical, 100% validated diagnosis. And we have to really look more towards how people are feeling and what their bodies are saying about things. So yes, I guess, but what would it take, in your opinion, for the more sensitive tests to become mainstream? Like why is that only available through blood banks? Why is that not something that is standard care for regular people?
I don't know the answer to that. It could just be that the perception is there's so little babesiosis that there's not a market that's perceived to be valuable. It could be as simple as that. One of the things that we're doing in our trials is defining cases based on the blood bank test and then doing other screens to try and see how often the other commercial tests pick up a positive case. And for example, we'll be working with Galaxy Diagnostics based in North Carolina. And the thing that distinguishes them from some of the other vector-borne testing labs is they've publicly committed to taking their assay through the FDA process. And that means that they're in a validation mindset. And I think a lot more of the diagnostic testing as it relates to babesiosis needs to be subjected to that level of rigor so that the we know what they can detect and can't detect. Where at least with Lyme, there are FDA approved tests, even though they have limited functionality. We're 10 years behind with babesia where Lyme is. And so part of it is just having other people active in the space saying there's a market here, and that's what we're doing. And we're hoping that some of the testing labs will come along for the ride.
Okay. And then maybe that's a good segue in the thought of how many cases are there of babesiosis out there? Do you have any idea? Again, CDC is constantly adjusting their infection rate with Lyme. And so I'm just kind of curious how that falls in place.
This is my prediction. I think we're going to see trend lines and institutional perspectives at CDC and elsewhere that look a lot like Lyme. So 25 years ago, there was hardly any Lyme, and now there are 900,000 cases a year based on administrative claims data. We know from our own research that we actually put out a press release today, which based on a study we did with Komodo, they're one of the biggest claims aggregators. And there are at least 25,000 cases of babesiosis a year where people are attempting to make medical insurance claims compared to the official case tally of 2,000 to 2,500 a year. So there's a tenfold difference just based on the hard data. And then we've also done patient surveys, physician surveys, and those data suggest the number could be into the hundreds of thousands of cases a year. And when you think about some of the data points on co-infection where you've maybe up to 50% of folks with long Lyme have a babesia co-infection. It wouldn't be that surprising if there's 900,000 cases of Lyme in medical claims that once the disease becomes more widely known, that you could end up with some significant fraction of that amount in terms of babesiosis claims as well. So that's kind of my take on it. And then the other thing is we just need more research. And it's kind of odd that it's a pharma company putting out the first report on administrative claims data for babesiosis. That should be something the CDC or the academic community is doing, but we have to do as part of our business case. So that's why we put it out there.
Yeah, well, I'm glad someone's willing to do it, and thank you for being that person, because in that company, it's just something that, again, really needs to become more top of mind for people, as opposed to just out of sight, out of mind, or just completely not knowing about it. Because again, if you're saying, I think I heard you correctly, that maybe 50% of people with long chronic Lyme have babesia going on. And I think about just in my small world of people that I help and how many people have been diagnosed with it. It's like 25,000, like that seems like a very small number in comparison to probably what it really is. And I just want people, again, to be aware that something like this exists, how it's transmitted to people. And I'm curious too, when I think about blood banks, I stay away from them just because I've had vector-borne illness, and I just don't think that they screen the blood adequately enough and I would feel terrible, but like I don't want people to not have blood either. So if someone's blood were to be infected with vector-borne illness, even things outside of babesia, is there a way for them to clean that blood to make it safe for people? And are they screening for all vector-borne diseases as scrupulously as they are with babesia?
Difficult question.
Sorry.
Yeah, no. So
Going a
I'll
little
I'll
crazy.
answer it based on what I know with babesia first. So the the official rate of transmission of babesia through the blood bank has dropped 95-98% since 2019. And that was the date this FDA approved test got introduced. Um, so I think if I was receiving blood from the blood bank, I'd feel pretty confident I wasn't going to get babesiosis. Um, I don't know what the standard testing panel is for other vector-borne diseases and whether the level of rigor is the same. I don't know. I'm not saying they don't do it. I'm saying I really don't know.
It's okay.
Yeah.
That's not probably more so in your wheelhouse, but while we were on that topic, I thought maybe you would know. And do you find that there are segments of the population that are more susceptible to vector-borne illness? I've had the thought that maybe it's a blood type thing. So like A positive people get bit by mosquitoes more readily or bit by ticks more readily than people with that are say like O negative or something. Do you know anything about that in your research?
Um, not specifically. I have seen some um, and I don't know whether it's genetic or environmental, but it seems like there might be a group of people who are more susceptible to um, chronic illness triggered by an initial infectious agent. So that the all I think about chronic disease as basically being a story of um, post-infectious syndrome caused by multiple different things. You've got long COVID, you've got long Lyme, you've got classic chronic fatigue, which can be triggered by numerous kinds of viruses. And then the thing we don't know is whether the infectious agent that caused those original infections, to what degree does persistence of the organism continue to dysmodulate the immune system? And and I and I think we're only just beginning to understand some of the mechanisms that cause the chronic syndromes, but there may be a common pathway, and it may be that people who've had those symptoms before are more prone to them, particularly if they had, like, for example, I have seen that folks who had more severe illness with COVID when they had the acute disease tend to have more severe chronic symptoms as well. And then there's the obvious, more exposure to ticks, you're more likely to get it. But I don't know that we have any more insight than that as to whether there's specific, immune molecules or blood groups or that makes you more susceptible to chronic illness.
Thanks. That's been a question in my brain for a very long time, and I'll get asked that by a fair amount of people actually. So just wondered if you had any more insight you could share on that. And curiously speaking, would you know like how many mosquitoes have Lyme in comparison to malaria?
So this is where we get back to what we say we know, and then what we don't know. So there's zero doubt that mosquitoes transmit malaria. I'm not aware of any literature that suggests that spirochetes can be, so the Lyme bacterium can be transmitted by mosquitoes.
Okay.
But I haven't looked very hard either.
Okay.
And so I don't know if my answer to your question is being dogmatic or factual. But that's what I know based on what I've read.
Okay, thanks. Because that's another thing I just think about with it being, more of a vector borne. I've heard people tell me they got Lyme from a wasp, from spiders, from mosquitoes, not just ticks. And so I was curious, is a mosquito the only thing that can spread malaria?
As far as we know, yeah.
Okay. Okay.
Yeah.
And then can you help me to understand a little bit more? What are some of the major differences between Lyme and babesia, especially more from a symptom set, or are they very overlapping?
They're different and overlapping, which of course is not very helpful and might be part of the problem. So obviously caused by a different organism. Lyme by spirochetes and babesia by a parasite that infects your red cells. So that means they're susceptible to different drugs. So doxycycline won't do much to eradicate a babesia infection, and atovaquone won't do very much to eradicate a Lyme infection. So there's that difference. In the early phase, they both trigger the same sort of flu-like illness, but then you have different manifestations. So with Lyme, you can obviously get a rash. With babesia, you can get anemic and be hospitalized with all the complications associated with a severe anemia, which doesn't happen with Lyme. And then, of course, when the Lyme organisms disseminate, you can have cardiac, neurological, and joint issues while the organisms are still dividing and multiplying and dispersing to those parts of your body. And with babesia, it is a red cell pathogen. And the thing where the science is really immature and we don't really understand is what causes it to persist. With malaria, there are well-recognized life cycle stages which are different and allow the disease to reactivate. But we don't know what allows persistence with babesiosis. And then with Lyme, there's 1,000 different viewpoints on whether there is a persistent organism, what does it look like, how do you treat it, and not a common set of facts or knowledge that allow even a discussion about, what the treatment guidelines for if you come from the starting point that there are residual organisms. So that's those, and of course, in the chronic phase, because they both do have a chronic phase of the disease, many of the symptoms overlap, and not all of them. And then at least what I've heard is you can get to recovery from Lyme, but if your babesia hasn't been dealt with, then you don't get to a full recovery. And the reverse might also be true. If you start out with babesiosis and then you haven't treated your Lyme, then you're not going to get to a happy place either. So that's kind of what I put together through, the literature and then talking to physicians and patients as well.
Okay. And I think that you're bringing up some very valid points in that it's very important for people to understand if they have one or the other or both so that they can get adequate treatment to deal with them, because they are separate organisms. They don't take the same type of treatment. And that's what I always try to express to people, is that Lyme testing is only looking for Lyme. It's not looking for the co-infections. And you really need to understand if you have those on board to get the adequate treatment that you need, because you could put all your eggs in one basket and just go hardcore Lyme. But unfortunately, you have like babesia or Bartonella hanging out, and you're not treating that properly, because again, it's a different type of path forward with each one.
Yeah.
How do you see, so kind of switching completely, but I also noticed that you deal with fungal diseases.
Yeah.
Does that play a factor in people when it comes to things like babesia and Lyme? Are you looking more at like mold when we're talking about fungal diseases, or what kind of fungus? Are you talking more like ringworm or what?
So our research with tafenoquine in fungal diseases started with we did a really broad panel of testing against bacteria, gram negative, gram positive, yeasts, and other kind of fungi, because we were just interested in seeing what other things our drug could be used to treat. And then we noticed that in some of the in some of the data that was transmitted to the FDA in the initial approval, that the levels of tafenoquine in the lung are the highest. They reach higher levels in your lung than anywhere else in your body following an oral dose. And so the question was, could we target lung fungi that affect the lung with tafenoquine because of the high drug levels in that particular organ? And so the first study we published, that seemed to be borne out in the animal testing, is that we reduce fungal burden in the lung specifically. And we're in the process of doing some other work to confirm that finding with a different kind of basically with Candida.
Okay.
And so, a plausible line of development, and I'll express this in terms of product development. A plausible line of product development would be to try and see if there are, important pathogens that affect mostly the lung, and could we put together a clinical trial program that would prove that's true. But the data for pathogens that affect other parts of the body are less encouraging, just because you don't get to high enough drug levels in the liver or the kidney, which is where you see a lot of hospital infections. So that's what we're focused on. And beyond the kind of preclinical testing that we're doing, the only other data there is out there is to treat a bug called pneumocystis, which is also a lung infection that it used to mostly affect HIV patients, but now mostly affects transplant patients where you have a suppressed immune system. So it's plausible that tafenoquine could be active against that particular pathogen. But that's where that's kind of where the science ends. We don't know much more than that.
Okay. Um, can you then also talk a little bit more about how people would stay safer? Is there a way, should they be taking tafenoquine here in the US to prevent things? Should they be taking that like once a quarter, once a year or something to tap down on some of the levels in people's bodies? Or no, it's not anything like that, and just do your best to put on repellents and stay safer that way?
Um, so this is a tricky question, and it's a tricky question because of my position, um, and what I'm allowed to say and not say. So I'll stick to generalities and then be very clear when I'm speculating. So when I go out in the woods, I always wear long pants tucked into my socks. And if I'm particularly worried, long sleeve shirt as well. Um, if I was hiking in Rhode Island, I'd be spraying myself up and down with DEET or with or doing some of the permethrin. Um, but I don't do that in DC where I feel like the risk is lower. Um, there, we have pretty good oral and topical, both tick killing, mostly tick killing medications for dogs. Um, and I think there's one company that's developing a similar kind of product um, for clinical use, but it's early in development. But that looks like a pretty interesting product when it becomes more mature. I suspect with babesia specifically, and now I'm speculating because there's no medical evidence yet to support what I'm about to say. Um, but I suspect, based on what we know about tafenoquine preventing malaria, that there's a reasonable chance that it would be the same for babesiosis. And at various times we've thought as a company about developing that indication for Arakoda and not treatment. But it's hard to do treatment studies, and it's even harder to do prevention studies. And so we you just need more subjects, and it takes longer, and that's why it's taken Pfizer so long to develop the Lyme vaccine. So then that gets to the next issue, which is Valneva, and Pfizer should be making some announcements. I don't know if it's the end of this year or next year about their Lyme vaccine progress. I would probably get vaccinated when the time came, because I feel like the risk of an adverse event is pretty low relative to the certain or near certain outcome if you actually get Lyme and don't get treated properly. So that risk benefit balance to me makes sense. And then I guess the other thing is and now I'm again expressing my opinion, and this is not a clinical guideline, and I'm not a physician, but if I had a physician tell me no when I had acute and I'd had an exposure to a tick and they wanted to test, unless you have access to a screening service that will test the tick in less than 24 hours, um, that's almost, for me personally, as a patient, bordering on malpractice. And I just feel very strongly that if you've taken the trouble to go to urgent care and you've been exposed to a tick and you've got those symptoms, the least harmful thing to do is err on the side of assuming that you've been exposed to Lyme and take the course of doxycycline rather than defaulting to a testing architecture which is only ever 40 to 50% effective in the acute phase of illness. And I think it's always going to be like that. I don't think we'll ever have a test for Lyme that in the acute phase will be better than 50%. And so why do physicians insist on testing in the acute phase of illness when by the time you get the test results back, you've got disseminated disease and all those problems? And again, just to disclaim everything I just said, I'm not a physician. This is me in my own capacity talking about my personal views.
Yep, I appreciate that, being clear on that, just being more of an opinion as opposed to actual fact. But you know, I always wonder for people, like, is there a better way to stay safer? And I preach it all day long, and tucking in your pants into your socks, long sleeves, shower within 2 hours from coming in from outdoors. But again, it doesn't really prevent, like, you could stick me outside and I just get devoured by mosquitoes. And I'm like, because what I've been trained is that you can contract Lyme from them. And my neighbor is so nice. He has a landscaping company, and he just gave me some new things that they're testing to prevent mosquitoes. But I've got them out now around my property because I'll try to stand outside at night and talk to them, and I can't because I'm just like, you know, like, why am I getting devoured?
Yeah.
And they're just standing there looking at me like I've got, you know, issues like that. Just they don't get bit and I get chewed alive. And so I just wonder for people, like, is there a way to stay safer? Is it smart to go for more of preventative versus reactionary when it comes to treatment? You know, really, what do experts think more so on that topic? So thank you for speaking a bit to that. Maybe an easy question for you. I'm curious to know why you call your company 60 Degrees. Is there a meaning to
yeah, so an equilateral triangle has 60-degree angles and it's perfectly balanced. And our goal is to find products that have a tropical country use and then a high income country use. That was the original mission. Now it's morphed into a more general focus on infectious disease and its consequences, but we still seem drawn as a company and humans to diseases that are really underserved, and babesiosis seems to be one of those. And but I think, you know, from a commercial perspective, there's I suspect the disease burden has been vastly underestimated, and I feel like we can do a lot of good there.
Yeah, and on that topic then, like, how do you get funding for your research? Because again, I know what it's like for people who are playing in the space of things that are not seen as they really should be. And like the funding is very challenging for Lyme disease research for one, but I would imagine it's probably more slim for something like babesia. How does that work for you? Are you getting
So I think we're very fortunate that we already have a commercial product. We're very fortunate that some pretty dedicated folks in the academic community took on the challenge of seeing whether our drug worked against babesiosis independently of us originally. And frankly, patients took a risk on a new medication and kind of told us how they were feeling and why they did it. and we are trying to get the same dose approved for babesia that's already approved for malaria. So what that translates into is that the number of clinical trials we need to do a small number of patients, just about the least risky clinical development plan you could do from a budget and success standpoint. And because we're focused on the commercial side with malaria and then repositioning that same drug at the same dose for a disease that looks very similar to malaria, it so far has been relatively easy to raise modest budgets to do modest trials. But if we were starting from a molecule in a lab that's never been into a trial and you have to go through that full pathway, we're not at the point yet where the babesiosis market's been proven out enough for someone to take that commercial risk that early in development. So that's one answer to the question. And then the other answer is just there's a lot of happenstance and luck in drug development, and we were fortunate enough to be able to license a product from the Army that had already been through 20 clinical trials. And so the overall capital investment and risk was okay for a small company. So that's the other way people do it, is have really strong partnerships with funders and you select projects that have been substantially de-risked. And then, unfortunately, the way the pharma system works is if you look into the future and look at product revenues and it's less than 200 million a year, the very few of the big pharmas will take the time or effort to develop a new product for something with, when you think about it, $200 million a year is a lot of money. But when you talk about most new antibody drugs get revenue into the billions, it's just very difficult for big pharma to justify the cost and risk in such a small market area. And then often by the time you get to the point where the risk it would be acceptable for them, they're really complicated projects. They've got, difficult product histories, and there are lots of partners involved and complicated data sets to resolve. And so it becomes, I think the phrase people use is it's got a lot of hair on it. And it's just difficult to pigeonhole it into a traditional pharma development campaign. So a bit of luck, bit of skill, and just focused on projects that have been de-risked enough to make it.
And so then I see that as a differentiating factor between your company and a traditional pharmaceutical company, and that you're willing to put it more towards something that, like, obviously you want to make a profit and you want to make a living, but you're more willing to play the game with something that isn't quite as profitable as some of the other major pharmaceutical companies. Is that fair to say?
I think it's so people don't start small drug companies and keep going for 10 or 15 years unless they're incredibly passionate and persistent. And so just as a human being, you have to feel like what you're doing makes a difference but there are lots of folks out there in the academic and pharma community that feel that way. It's just there's not the opportunity for everyone to be able to do it. So that's part of it. And then, we will be profitable within the constraints of the disease space we're working in, because we don't need a $500 million a year budget to keep churning out new blockbuster drugs. We've got a much lower personnel and kind of capital footprint, and so you can make it work with a smaller market. But again, all the things have to align to make that possible.
And as a final question for you, I apologize. I know we're going a little bit long here for you on time, but what would be the direction that you see your company going in from here forward? Will you expand more into other vector-borne illnesses? Will you stay focused on your mainstay of babesia, fungal diseases, malaria, and dengue fever? Or again, will you change your scope a little bit more?
So our next clinical trial, I hope, will be in chronic babesiosis. And one of the entry criteria will be fatigue for 6 months or more. And I'm hoping that will be our first foray into providing a product for chronic illness that will be, make a difference. So that's sort of the direction we're heading in. One of the things that we did recently was, and we did this through two different ways, one through claims data and then the other through a 6,000 patient survey, or 6,000 US adult survey, so representative of the census. And both those data sources suggest that at any one time in the US, there are 10 million people who have experienced chronic fatigue for 6 months or more. And that's kind of not differentiating whether what caused it was long COVID, long Lyme, chronic fatigue. It's just asking the question about the symptoms people feel. And those 10 million people have no answers. There's no specific FDA approved treatments for post-infectious syndromes. And I feel like that's a vast unmet medical need that if it was 10 million people unaddressed in any other disease area, you'd have pharma companies all over it. So I think that's the future. I hope it's us doing a small part of that. Yeah.
Yeah, thanks. And I'm sorry, I lied. Is there a
One
way
more.
a way that listeners can help you? Is there anything that people in the general public could do to support what you're working on?
It would be great if you could buy 60p shares.
Okay.
And then we do have if every Lyme patient bought 100 shares of SXTP common stock, it would be awesome for us.
Okay.
And then we have a chronic babesiosis study that needs a name. And I believe we put out some some social posts about that, trying to find a formal name for the study. And we can reach out and provide a little more detail about that.
Okay.
And then when we get to the point of opening up our chronic babesiosis study for enrollment, we'll be recruiting patients to that study to evaluate Arakoda. So,
all right. And do you have a waiting list for that?
not at the moment. It's officially listed as no, is the short answer to your question.
Okay.
but I hope that we'll have some announcements about which organizations we'll be partnering with to do the study in the next couple of months.
All right. And would people check your company website, or where would they?
Yeah.
Okay.
Yeah. And then, of course, when we know what we're doing and when and how, we'll be pretty communicative through press releases about that, and then in podcasts when the opportunity arises.
Okay. And so for the listeners, if you're interested, you can go to 60degreespharma.com
Yeah.
and keep checking back for information on that, because I'm sure there's people who are listening, especially those that are infected, that would probably be willing to take a chance and participate if they've been struggling for a good length of time with figuring out how to help themselves. So thank you for that. I appreciate it. And I really do appreciate your time. I know we've again gone here a little bit long, but I have still like 50 more questions that I could ask you. Stuff is very fascinating, and I appreciate your openness and also your humanness in stating that some of these questions that I've asked you, you don't know the answers to, and just doing your best to navigate through them, because those are the difficult ones when you're really not sure of the answer specifically, but it is something that you're passionate about and that you care about and that you want to understand better so that you can make your mark on people and help them truly get through something that can be rather debilitating. So I appreciate you very much. Thank you.
Thanks a lot, Annie, and I'll talk to you again soon.
Yes, I hope so. Thank you very much, Dr. Dow.
Okay. Thanks. Bye.